So,
Good
good
morning
morning to
to
all
all
of
presenting here. I am Sourav Ghosh from India, working as UGC Senior Research Fellow in University of Kalyani. Now, I am going to deliver a lecture on the topic Anti-biofilm activity of nanonized eugenol. Now, a common question has been arised in you. What is biofilm and why is it? Actually, bacteria alternates between two states. One is motile planktonic bacteria, another one is surface attached sessile multicellular community called biofilm. Biofilm is a multi-step phenomena which is formed by different steps such as surface attachment, microcolony formation, and then mature biofilm formation. Biofilm is a bacterial community which is enclosed by self-produced extrapolymeric substance which protects the biofilm residing cells from the action of antibiotic as well as antimicrobials. Biofilm causes most of the chronic infections. As per report of WHO 2017, biofilm-mediated antibiotic resistance mortality at about 7 lakh people globally per year. Some of the clinically relevant biofilm-forming pathogens are Acinetobacter baumannii, Staphylococcus aureus, Staphylococcus epidermis, Vibrio cholerae, and Pseudomonas aeruginosa, out of which Pseudomonas aeruginosa was selected as target organism for our study. But why? Pseudomonas aeruginosa is an opportunistic pathogen. It causes various nosocomial infections like bacteremia, sepsis, urinary tract infections, etc., especially in immunocompromised individuals with cystic fibrosis, burn wound, and organ transplantation. Its global mortality rate is about 13 to 50%, and who considered the pathogen as greatest public health threat. That's why its treatment strategy is needed urgently. We have to find out such kind of treatment regimen which should have both the capabilities of inhibition of biofilm formation as well as eradication of pre-established biofilm. Inhibition of biofilm formation strategies include retardation of surface attachment, microcolony formation, and killing of biofilm forming cell so that each and every steps of biofilm formation can be prevented. Another strategy is development of anti-fouling agent so that we can get an effective coating agent for biomedical devices to inhibit the bacterial colonization. Another way of strategy is eradication of pre-established biofilm, which include degradation of EPS so that biofilm residing cells are exposed to the antimicrobials, and killing of biofilm resident cell to stop further infection at different sites. All of the possibilities are available in one compound, that is plant essential oil eugenol, which is majorly found in basil leaves, clove bud, cinnamon, etc. Now, the time to evaluate eugenol as drug candidature. Eugenol has some advantage as well as some disadvantage also. It has versatile pharmacological profile including antimicrobial, analgesic, antioxidant, anticonvulsant, neuroprotective action, etc. It is green in origin, that's why it is less toxic and not harmful for nature. It is easy to decay, and the demerits of eugenols are it is deeply water soluble, that's why it is less bioavailable to our systemic physiology, and it is less stable also. Therefore, what will be the solution for eugenol? Yes, you are correct, that is nanonization. Eugenol was nanonized by ultrasonic cavitation. It is simply emulsification of hydrophobic eugenol with hydrophilic gelatin by the process of ultrasonication. Some of the physicochemical characteristics of eugenol nanoparticle are mentioned here. such as hydrodynamic size, pore particle size, polydispersive index, entrapment efficiency, and release profile of eugenol from gelatin core. Anti-biofilm study of nanonized eugenol was performed by various parameters such as determination of MBIC, that is minimum biofilm inhibitory concentration, and MBEC, that is minimum biofilm eradication concentration. Another parameter is biofilm biomass, that is corroborated to bacterial attachment on surface, pellicle, that is bacterial attachment on liquid interface, EPS, which provide protection, biofilm thickness, swarming, that is related to EPS secretion, and twitching, that is correlated with microcolony formation. MPIC is defined as a lowest concentration of any drug which causes more than 90% inhibition of biofilm formation. Inhibition is measured in terms of bacterial viability. Eugenol nanoparticle shows more than 95% inhibition here, as well as MPEC is defined as the lowest concentration of any drug which eradicates more than 90% of pre-established biofilm. And in this results, eugenol nanoparticle eradicates more than 90% of pre-established biofilm. Biofilm biomass is determined by surface attached cell staining with crystal violet. The intensity of blue color is directly proportional to biomass. First panel is showing the results of biofilm inhibition, whereas the second panel is showing the results of biofilm eradication. In both cases, eugenol nanoparticle exhibits better results than that of eugenol. Pellicle is a form of microbial assemblage which is formed at air-liquid interface. It is more clearly visible in glass tube than well plate. For assessing pellicle inhibition and eradication, same experimental method was followed as biofilm biomass. In both cases, eugenol nanoparticle is showing better results than that of free eugenol. EPS in biofilm architecture is composed of cellular polysaccharide in major and DNA in minor amount. The dye congruent binds EPS and the intensity of red color is directly proportional to the quantity of EPS. Eugenol nanoparticle exhibits better EPS inhibition and eradication than that of free eugenol. In this study, the fluorophore cytonine is used. The biofilm thickness was measured by jet stacking method in confocal microscopy. The pictures of this slide are clearly showing that eugenol nanoparticle exhibits better action in the inhibition and eradication of biofilm thickness than that of free eugenol. Bacterial swarming is totally dependent on flagellar functionality, and it is correlated with EPS secretion. Swarming efficiency was assessed on dry agar plate by measuring the diameter of zone of movement on agar, and the images shows that eugenol nanoparticle exhibits better action, that is better swarming inhibition than that of free eugenol, whereas bacteria twitches on solid surface by successive extension and retraction of surface filament known as type 4 villi. This aids in the surface attachment, that is first step of biofilm formation. Eugenol nanoparticle is also capable to inhibit twitching phenomena significantly than that of free eugenol. To establish eugenol nanoparticle as coating agent for biomedical devices, bacterial colonization was studied on nanoparticle coated and uncoated catheter. On uncoated catheter, bacteria forms a thick bacteriofilm, whereas on eugenol coated catheter, there were few number of bacteria found to be attached, and on eugenol nanoparticle coated catheter, there were negligible number of bacteria were found to be attached. In another study, cellular morphology was studied to investigate the antibacterial mechanism of eugenol nanoparticle. Control cell exhibit rod-shaped perfect structural cellular integrity, whereas eugenol treated cell exhibits the blistered rod-shaped cell along with some protrusions on cell membrane as spheroplast, while eugenol nanoparticle treated cell exhibit complete disruption with appearance of cellular fragments. This is summary slide. Our prepared eugenol nanoparticle is capable to inhibit each and every steps of biofilm formation, and when it is applied on mature biofilm, it degrades the EPS component of the biofilm and kill the bacteria by bursting the cell. Eugenol nanoparticle is also used as effective coating agent for catheter, and bacterial colonization was prevented significantly on coated catheter. Now the time to conclude our work. Our synthesized nanoformulation is very easy to prepare. It's a pure organic formulation, that's why it's less toxic and not harmful for nature, and the cost of this ingredient are very less, that's why its manufacturing cost is very low, so that it can be afforded by every group of people in our society. It is also used as anti-biofilm agent. It is also grouped as antibacterial coating agent for biomedical devices. And our proposed antibacterial pathway or antibacterial mechanism of our prepared nanoparticle is about membrane blistering that leads to bubble protrusion, that leads to spheroplast, and that leads to burst the cell. I'm happily announcing that my work has been patented, and the patent application number is mentioned here. Our future plan are determination of amelioration potential of eugenol nanoparticle in mice on the basis of bacterial count in lung homogenate and BAL fluid, that is bronchoalveolar lavage fluid, histopathology, and the determination of level of cytokines such as IL-6, macrophage chemotactic protein MCP1, IFN gamma, TNF alpha, etc., histochemistry, and all other experiments. Besides this, we also determine the anti-biofilm activity of eugenol nanoparticle in clinical isolation. This is acknowledgement list. We want to acknowledge UGC Government of India for funding fellowship, DBT Government of India, RUSA Component 10, DST Government of India, and the University of Kalyani. They are my lab members. Taruk Das Basu is my supervisor, and the other members Upasana Shet, Sanchita Nandi, Pothikrit Bandyopadhyay, Somojit Chakraborty, Anabhatya Pal, Sushmita Nandi, and Obhijit Das are my colleagues. Thank you all. We are willing to collaborate. If anyone interested, please drop a mail to the following email address. Thank you again for your patience.